here is 30 seconds on how to do molecular biology. You get some sequences. I don't know how you do that. That's wet lab type stuff. You do an alignment. That's kind of my department. Then you do some analysis, and then you publish. So this is just enormously widespread in molecular biology. I mean, muscle gets, I think, 10 citations a day now, and it's not even the most highly cited method. So this is something that people do all the time. And kind of the de facto standard for what people do is they kind of use whatever their preferred alignment method. It might be muscle or some other program. They might try and sort of adjust it by hand, but whatever you do, you kind of proceed on the assumption or the hope that the alignment you have is good enough for whatever analysis you want to do, or that whatever problems it has, you can deal with true tree bootstrapping or something like that. But there's generally sort of it's kind of a gaping hole in the field, really. It's that we rely on alignments a lot, but for the most part, we don't know how good they are, and we don't really have statistical methods to deal with any errors that there might be in the alignment. And that's what this method I'm going to describe today is designed to address. So just to give a bit of background, maybe you're not so familiar with actually using alignments in practice. So this is 4 proteins. These are real proteins. And here I'm showing 2 different alignments of the same sequences. And so, well, which one of these is better? Is either of them correct? Is one of them better than the other? And the sort of the dirty laundry of the multiple alignment field is that there's really no way to decide. Even if you have, in the case of these proteins, we have structures, but even with the structures, it's very hard to say. So in the green region, it's very well conserved, the sequence similarity is obvious, the sequence homology is obvious. If you look at the structures, that green region is a well-conserved anti-parallel data sheet. So it's pretty much unambiguous, except maybe for one gap. So we're pretty happy. But if you get into the red zone here, then it's a completely different story. The sequence lengths are quite different. The structures are quite different. Here we have probably a surface loop there, which is just, it's going in a different direction. It's probably doing different things. And it's quite possible that the sequence homology has been completely lost. You can get sort of repeated cycles of insertions and deletions and little tandem duplications, whatever, and at some point it just becomes meaningless. There is no homology anymore. So should we even be trying to align that region? We don't know. So what I'm suggesting is that we can deal with this by making replicates, by making a lot of different alignments that are equally plausible or equally accurate as far as we can tell. And then we can ask, are the conclusions that we draw from this alignment, the tree, the enzyme classification, whatever it is, are those conclusions robust when we try it on all of the different members of the ensemble? So how do you go about making such an ensemble? So let me start with a sort of simple toy example. So you're probably familiar enough with alignment algorithms to know that they use things called affine gaps. So you have sort of a substitution matrix which gives you a score for every pair of letters that you might put in a column. And if you open a gap, you subtract a penalty. And then if you make the gap longer, you subtract a smaller penalty. And so, for example, in BLAST, the open penalty is 5, the extend penalty is 2. But there's no sort of magic to those values. I mean, they're probably tuned on some benchmark. That does it really matter if its open was 5.1 and extend is 1.9? Well, on average, no, that shouldn't matter, because they're really not very sort of, as I say, there's no sort of very magic, well-defined values that they should take. So the idea is to perturb the parameters. Let's just introduce some small random variations and see what happens. And the idea is to sort of fix the size of the variation and then pick some random values from within that range. And because if the changes are small enough, it really shouldn't change the average accuracy of the alignment. So what's going to happen when we try an alignment like that green region? Well, it's going to be robust. I mean, we've hardly got any gaps in this case, or it's very clear where the gaps should go when you have similar sequences. So the alignments don't change very much. So that's my little jargon there, is dispersion. That's kind of the average distance between the alignments in the ensemble. So if the alignment's robust, then we have small dispersions. I mean, it's not uncommon that all the alignments would actually be identical and then the dispersion is zero. On the other hand, if you do the same variation in parameters, like with the red, that red region, you may find that you get lots of different alignments out when you make small changes to the parameters. So this is what I call large dispersion, and that's kind of a sign that you should, the alignment may not be very robust or very meaningful, and this is where you start to worry about whether your downstream analysis is robust or not. So that was like a simple toy example. So what do I actually do? So what I actually do is I make alignments with this hidden Markov model, which is a rather big and complicated thing. I can't really go and explain this whole idea in this talk, but to give a quick summary, it has affine gaps, but it actually has two sets of penalties. It has one pair of penalties which models short gaps, and then another pair of penalties for long gaps. So for the long gaps, the open penalty is higher, but the extension penalty is lower. And it just turns out that this works well in practice. And then we have a matrix for amino acids is going to be 20 by 20, which gives you roughly 200 independent values. For nucleotides, it would be 4 by 4, which is, like 10 or something independent values when you consider the diagonal. So altogether, we have maybe a couple of hundred probabilities that we can perturb and see what comes out of the alignment. So here's the math. You take each probability and you kind of bump it up or down by a random value up to some maximum. And the maximum is set by the amplitude. That's the alpha parameter. And delta is a uniform random real number which varies between -1 and +1. So this basically gives you plus or minus 25% on all the parameters. And then we have to renormalize after we've done that to make sure all the probabilities sum to 1 and they're not greater than 1. So there's another way that you can run into trouble with alignments, and that's you can get bias because of the way the alignment is built. So in practice, just about all practical multiple alignment algorithms, muscle, T-coffee, probcons, clustable W, clustable omega, at some point they all do this thing called progressive alignment to get the final result. And the way that that works is you have a binary tree, and at the bottom of the tree is the unaligned sequences. And at each node of the tree, you align the subtrees on the left and the right to build a new MSA, and you keep the subtrees fixed. You just introduce new gaps in order to align those two subtrees. And when you have challenging alignments, you can see that errors and gaps necessarily increase on a path from the leaf towards the root. So if you imagine what happens at one node, over on the left, you can see, well, we've got some columns that would be kind of like the green area, things are working well. We've got other columns which are like the red area. Here I've shown them in orange. They're meaningless or kind of filled with errors. And when we sort of combine those two, whatever errors we have so far, well, we're stuck with them, and we're going to introduce new errors and get new meaningless regions in the combined alignment. And the net result of this is that if you look at sequences in subtrees from the guide tree, they're going to align to each other better than other subgroups. And this means that if you give that alignment to a maximum likelihood program like RAXML or IQ tree, then you should worry that the topology that RAXML is going to estimate is going to follow the guide tree, because it's seeing those patterns which are really artifacts of the progressive alignment. So this is basically a picture to say in what I just said in words. So the maximum likelihood tree or the parsimony tree, whatever, may recapitulate the guide tree, and that's just an artifact of the way that the errors and gaps accumulated. So this suggests another way that we can make variant alignments in our ensemble. Well, the idea is to use different guide trees so that if there are biases and there is this kind of effect that I just described, they're going to be distinctly different in each of the variants. And the way we accomplish that is by permuting the joining order of 3 large subgroups. It's actually quite tricky to achieve in practice, but that's the sort of conceptual idea, and the details are in the paper. If you don't want to shuffle the joining order close to the leaves for a couple of different reasons. One is the effect's going to be small because you're only, moving around 1 or 2 sequences. And the other reason is that you want to maintain accuracy. The idea is that any variant coming out of this procedure should have state-of-the-art accuracy. Otherwise, well, you're not going to use it. You're going to explain any differences you see because the alignment is inferior. So it's important that you keep the joining order close to the leaves so that you're aligning closely related sequences, because those are easier to align more accurately. So here's kind of a couple of different ways you can arrange the guide tree, and this is to kind of suggest the notation you're going to see in a minute. So one of the permutations is called ABC, that's kind of the default. Then we've got this other one, CBA. And here's a final way that you can make replicates, which is actually quite useful in practice. So sometimes you may have thousands of sequences, and that's too many to do certain things. So for example, RAXML, if you give it a few thousand sequences, that it just can't handle it. So one of the things that you can do is you can cluster those sequences and then select one sequence from each cluster at random, and then you can make an alignment from the subset. Well, you can choose different representatives from each cluster, and this gives you another way of making alignment replicates. And what you hope to see is that the joining order, or whatever it is that you're doing, is consistent with the when you infer a tree. But if they don't, then you know that maybe something is going wrong. It's giving you a sort of a consistency check, a reproducibility check. So what kinds of ensembles can you make? So this is one that I like to use for sort of doing visual review. And it's kind of a 4x4 matrix. And on one axis, you keep the guide tree fixed. And on the other axis, you keep the HMM parameters fixed. So we have the same gap penalties, the same substitution matrix. And that way you can look at your downstream predictions. So are you predicting an enzyme category, secondary structure, a tree? You can kind of visualize that in the 4x4 grid and see whether your prediction is robust against the variations on each axis. Another thing you can do, this is useful for bootstrapping, is you try and maximize the variation between the different alignments. And so we don't reuse any of the HMM parameters. So every replicate is using a different HMM, and then we just cycle through the 4 different guide tree permutations, which the algorithm uses by default. And so here is sort of the typical way you do it, is you do a 25 by 4 matrix, and that gives us a 100 replicates for bootstrapping. And one of the nice things that you can get out of the ensemble is you can get a confidence measure on a column, which is similar to the bootstrap confidence on a tree edge or a tree bifurcation. And the definition is the same. It's the fraction of replicates where you find that column. So you can see here we have a trivial ensemble again with just 2 replicates. And you see the very first column, well, it's reproduced, it's perfectly conserved, and it's got a confidence of 1.0. But if I look at the first column in the red region, you'll see it's not reproduced. It's only in one of the 2 replicates, so the confidence is 1/2. And so here is sort of a slide with too much text and notations, but this is basically this is a short description of the standard method of doing tree bootstrapping. And the idea is that you make 100 replicate alignments. Now, this is a different kind of replicate from everything I've been talking about up to now. It's you start from your one favorite alignment, and then you build an ensemble by resampling columns with replacement. Then you make a maximum likelihood tree, or whatever it is, whatever kind of tree you like from each replicate, and then you assess the confidence of an edge by seeing how often that edge is reproduced in your ensemble of trees. And there's a modification of that procedure which was recently proposed. There's a Nature paper from 2018, and this addresses the problem that when you get many sequences, you often don't reproduce an edge, but you get very similar subtrees. The problem with the standard Felsenstein method is that it only takes one difference and the edge isn't reproduced anymore. So the idea with transfer bootstrap is to look at whether an edge is approximately reproduced, and you get some credit for that. And this method was used in a study that I'm going to be referring to, so that's why I'm giving the background here. And I'm proposing a modification of the standard tree bootstrapping, where instead of sampling columns from a single alignment with replacement, you sample columns from all of the replicate alignments, and then you make a maximum likelihood tree from each of those. When the dispersion is 0, when the alignment is robust, all the replicates are the same, and then this is exactly equivalent to the standard Felsenstein bootstrap. But when you have an unreliable, less robust alignment, notice that columns with high confidence are more likely to be sampled because there are more of them in the ensemble. So the resampled MSA that you're using for bootstrapping has more accurate site patterns on average, and you're going to get more accurate trees in your ensemble. So hopefully you get better bootstrap values. That's what I just said. And conversely, so think about what happens in the sort of the conventional Felsenstein procedure. So you make a alignment, and it comes out of muscle or whatever, and you tend to believe it, but actually there is systematic bias in that alignment due to the guide tree. So when you make the ensemble with guide tree permutations, you're going to get different branching orders because now your bias is in a different direction. So in that case, when you do the bootstrapping over my type of ensemble, you're going to get lower bootstrap errors, which lower bootstrap values on edges which are due to the systematic error. So this kind of procedure intuitively seems to have nice properties. You're going to get higher confidence values when edges are good, and you're going to get lower confidence values when edges are due to systematic error. Well, that was all nice theory, but can we do some results in practice? So first, let's look at the overall accuracy. So the blue, okay, what's the y-axis here? We're looking at protein alignment accuracy on the Barley-based benchmark. So that's kind of the de facto standard benchmark for multiple alignments. And one of the things to note, if you're not an expert in this sort of area, is that even the best methods only get around 50–60% of the columns correct, which means that it gets 40 to 50% of them wrong. So this whole concern about, well, maybe your alignment isn't robust, that's a real thing out there when you have challenging alignments. This really is a practical problem to worry about. And you'll see on the left, those blue bars, that's a state of the art, that's Plustal, Omega, and MAFT. And you'll see that muscle 5 with the defaults, that's the fat red bar, has higher accuracy than the state of the art. And what's also very important is any of the replicates, you can perturb the guide tree, you can perturb the HMM parameters. It's essentially the same accuracy. I'm not degrading accuracy by making these variants. So there's no reason to prefer the default parameters over anything else. And the only way to explain differences between these replicates is errors. There must be alignment errors there. And because you can't prefer any one of those alignments, unless you've got independent evidence from somewhere, then you basically have to assume that any randomly chosen member of this ensemble it's mostly errors. The differences you see with the other alignments, they're mostly explained by errors. They're not explained by the alignment being inferior. So if I do the same thing on nucleotides, we see a very similar story. This is Barley base, and now the R stands for RNA. It's really the only structural benchmark I know of for nucleotides. So if you remember, dispersion is the measure of how much difference do I see between different members of the replicates. So if the dispersion is 0, the replicates are all the same. If the dispersion is high, there's a lot of differences between them. And you'll see dispersion correlates quite well with error. So dispersion, it's unsupervised. In other words, I don't have the correct alignment available. So you can see already dispersion is giving me a useful informative prediction, which is, well, how much can I trust my alignment? If I'm seeing high dispersion, then I can be pretty confident it's got a high error rate. And if you remember the column confidence, that's the fraction of replicates where a given column is seen. So that's very similar to tree bootstrapping. And you can see here on the y-axis is the probability that a column is correct. And the x-axis is I binned all the columns in the benchmark into sort of ranges of 10%. So you can see if the column confidence is 100%, then it's sort of better than 95% probability that the column actually is correct. And the value is, it's highly predictive, it correlates well, but it's way over optimistic. So if the column confidence is 90%, then it's actually slightly under 50% likely to be correct. And why does it underestimate? Well, it's sort of a technical discussion, but one of the things to keep in mind is that the hidden Markov model simply can't generate most alignments. That's true of any automated alignment algorithm, they only, even if you change all of the gap penalties and substitution matrix values as much as you want, most alignments you just can't reach. So we're only exploring a small fraction of the possible alignment space, and it's quite possible. In fact, I would say it's probably certain in challenging cases that the correct alignment is just not reachable. So, but it doesn't really matter, because once you've got a benchmark, you can correct for this effect and you can get a column confidence, which is really a probability that the column is correct. So getting back to my little illustrative example here. So now we're basically looking at the same figure, but now we've got some background to understand more of the details. So you can see over on the right, those are the PDB structure identifiers for the structures that we're talking about. At the top is the column confidence values on a scale of 0 to 1. And that's measured on a diversified ensemble of 100 replicates. And I'm showing two alignments from that ensemble. One is the default HMM. There's nothing special about that, remember. It's just as accurate or as bad as any of the other replicates. And then, just as an illustrative example, the second alignment is with Permuted guide tree in the order BCA, and we have permutation C2, that's a random number seed, and you can see the alignment comes out quite differently. So here is a comparison of bootstrapping using a single alignment versus bootstrapping from the ensemble. And this is done on, where was this done? I think it was done on something like 10 PFAM domains. And you can see that the correlation, so what we do, so the experiment here is we make the ensemble and then we generate trees with RAXML, and we ask, and we look at the bootstrap value on each tree edge, and we ask, how do the two bootstrap values compare? And they correlate very well. And you'll see from the little inset histogram, you can see that the dots kind of tend to be in the bottom right corner. And that means that the ensemble bootstrap tends to give somewhat higher values. And if you remember that, that's the effect we expect if the errors are random. If the errors in the alignments are random, then doing the ensemble bootstrap is going to increase the confidence value in correct edges, because now we have more reliable columns. So that's how I would like to interpret that result. This is the same thing with transfer bootstrap. It's essentially the same result. And this is looking individually at the PFAM domains where I got that result. Now, the capital D inset in those scatter plots, that's the dispersion. So one of the things to notice here is, if you look at the bottom right, that has the highest dispersion. So the alignments are the most different in that particular domain. But that didn't seem to affect the tree inference. So you'll see most of the bootstrap values are right up in the top right corner. That means the bootstraps are close to 100%, the trees very, very little. And that tells you that the tree is not sensitive to variations in multiple alignment. And you kind of see the opposite effect in the top right corner. You'll see, well, the alignment is pretty robust. So we have a lower dispersion, but the trees are much harder to infer. You'll see we have bootstrap values going all the way down to 0. So here we have a different, sort of qualitatively different case. So what this methodology is doing for you, it's telling you how robust the tree is to uncertainty in the alignment, independently from how certain you are in the tree, given that you're assuming the alignment is correct. So we're able to untangle those two sort of uncertainty contributions. So now into a case study of one particular tree that I became seriously obsessed with for reasons I don't have time to go into here really, but this has been a highly influential study. So back in 2018, RNA viruses, such as coronaviruses, they're the sort of the primary virus pathogens for mammals. They were only classified up to sort of around species and genus because it was just considered too hard to unravel the phylogeny of these viruses. And then this paper came out in 2018, and they came up with this tree with these very high transfer bootstrap values. And then since then, this has been adopted by the sort of the official taxonomy organization of RNA viruses, and it's been used to define phyla and orders and classes and whatnot. And when I saw this tree, I was very skeptical because it's enormously difficult to align. So I should say that the sequences are polymerases. They're the polymerase that every RNA virus has, and it's a very difficult protein to align. It evolves quite quickly, and the diameter of that tree is something like 10 substitutions per site. So we're claiming that we can look back over these very long branch lengths and resolve very small differences in sort of height between these groups, and I found that difficult to believe. So this is a stratified ensemble of trees. And so what I did was I took those same sequences from that study and I realigned them using a Muscle5 ensemble. And you can see we have 3 different guide trees. The top row, it's the default guide tree, and then we have 2 permutations of it. And then from left to right, we have different HMM perturbations. And the edges are colored with what are now, you know, the currently accepted phyla for these guys. And you'll see the good news, at least if you're Wolf and company, the good news is that the phyla seem to hold up, which actually was not what I was expecting. You can see the reds and the greens and the blues, they lump together pretty well in all of these replicate trees. So we're kind of supporting the idea that these guys are in fact monophyletic, but we do not reproduce the branching orders. You'll see that each of these has a quite different branching order. To make that easier to see, I've got these sort of stylized trees which just boil down to the topologies. And on the left is the tree from that study with the transfer bootstrap values that they got. And on the right are a couple of the examples of the trees that I got with my transfer bootstrap values using Felsenstein resampling. So now I'm taking one of the replicates and I'm using the standard Felsenstein procedure, and what comes out are these high bootstrap values. So I reproduce the fact that we get high bootstrap values, but I get different topologies. So these can't all be right. So one conclusion we can certainly draw is that you can get high transfer bootstrap values for wrong trees. Now, if we repeat the exercise using the ensemble resampling, you get low support values. So my interpretation of this, it's kind of the opposite of what we saw with PFAM. Now we have, if we go back and look at any of these trees, I think we're getting high bootstrap values because the maximum likelihood trees are biased towards the guide trees that we use to make the MSA. And then when you bootstrap over and you can see, you know, the ensemble is giving different topologies. So when you bootstrap over the whole ensemble, you're sort of averaging over the guide tree bias, and we're getting much lower bootstrap values, which are, you know, too low, obviously, that you would believe the tree. So obviously the evidence has to be circumstantial because we can't go back 2 billion years and see what these viruses were doing. But I think the circumstantial evidence strongly supports that the ensemble bootstrap is doing what we would like it to do, which is to improve confidence in correct trees and reduce confidence when the trees are biased towards the alignment. So that pretty much does it. Thank you very much.
Thanks, Robert. That was a terrific talk and actually really well timed because we've been, in the age of the big sequence data, what we think is due diligence is just to look at the trees. And in some of the projects, we've been peeking back at the alignments and realizing exactly what you're seeing here, how unreliable they are. So this is perfectly timed. I'm going to open it to the floor. I might just start with one quick question, which is this in principle looks like a much improved approach, but how does it scale? And the second question together with that is how does it handle concatenated protein alignments, which a lot of the species trees are based on. And we know, for example, that standard bootstrap values grossly inflate with concatenated protein alignments. So how does it go?
Right. I'm very glad you asked about scaling because I should have said something in the talk. So ProbCons has been around for a long time. This is based on the ProbCons algorithm, and it's always been the top scoring alignment, this top scoring algorithm